All right. Today we are going to talk about gene drive. So, as a segue into that topic, we've spent a lot of time in this class talking about how to make transgenic organisms. And there's sort of a problem with all transgenic organisms. And let me draw that out for you. So, imagine you got a transgenic fly, okay? And this fly does something for you, function X, which you like, you're a fan of, okay? The problem of all transgenic insects is how do you get that genotype, that transgenic genotype, how do you get that genotype into the wild? Because you can imagine there's a scenario where this is the genotype you want. Maybe this is a certain mosquito that doesn't spread a disease, or maybe this is a certain insecticide susceptible pest, or maybe this is some insect with a favorable phenotype conveyed by a favorable genotype. How do you get that insect to invade a natural population of insects and eventually replace them? So this is what would be called population replacement, right? Like we have all these fancy studies in the lab building really, really fancy things, and the fancy organisms in the sense that they do all these things for us. How do we get those into the wild? It's different for agriculture, right? Like if you build a fancy plant, okay, that's easy for agriculture because they don't have to replace any plant. They just set up a field now with a bunch of those fancy plants and they do their harvest, okay? So it's different for agriculture, but now imagine the scenario, like I said before, imagine the scenario where you're trying to replace a certain pest population which is causing you a lot of problems, and you build this transgenic organism to replace them. How do you get that to invade the population and replace it? That is sort of the problem of many of the transgenic systems we work on. And there's a solution, which we're going to talk about today. The solution is called gene drive. Now let me just elaborate a little bit more on this problem of the transgenic organism. So again, imagine we make our transgenic fly by, let's say we inserted gene X, which prevents it from, it doesn't, how do I want to say this? It causes it to not spread a disease. Let's say it's a crop disease, okay? And this was conveyed by an insertion. Let's say we use a transposon to insert gene X, which inhibits the spread of some virus, okay? So this organism is transgenic for gene X. Now, there's an evolutionary cost to that insertion, okay? That organism, that transgenic organism, has to spend energy. That gene X had some promoter. It's got to spend energy, okay, to transcribe gene X and translate gene X into protein X, which does some function. And the cost of that function, or a couple functions, is there's a cost to those in the organism. Maybe there's a downstream metabolic cost, okay? So oftentimes we're thinking about what evolutionists, I guess, would call fitness. Many times when we create these transgenic organisms by inserting gene X, we are decreasing the fitness. And I guess fitness can in many ways be an abstract term. Let's imagine a scenario that would talk about fitness. Let's imagine a scenario where we were to compare the fecundity, which is the number of children, number of offspring. That's the fecundity. Let's compare the fecundity of a wild type, WT, to the gene X transgenic type. And let's say wild type fly has 100 babies on average. And the gene X transgenic baby insect has 75 babies. This one is going to be significantly less fit because this one has 25 extra babies. And maybe that's conveyed by because it doesn't spend extra energy on making protein X. And the function of protein X in the wild type, it's not there. We haven't inserted the protein X, so there's no downstream cost. So maybe this one just does better and has more babies. Okay? This is going to be a huge disadvantage, and this one is going to be selected against. And this one is in theory going to be selected for, because this insertion is actually decreasing the amount of babies, decreasing the fecundity of that organism. So this is a big issue in transgenics, is what's the cost? What's the fitness cost of inserting these transgenes? And in many cases, this is actually the scenario where in many cases, they don't do as well. They don't survive as well. They're not as fit. They don't have as many children or offspring. And so it's very, very difficult to sort of release them into the wild populations to replace them, which is what needs to happen if you want your transgenic insert with gene X to have some kind of an effect on the population as a whole. Okay, let's also talk about and review some Mendelian genetics to help you sort of also see another reason why transgenics have a difficulty in invading populations. So let's draw a Punnett square, if you remember from basic biology. And imagine that we're discussing flies, okay? And flies, we're talking about a transgenic fly, which is diploid. And let's say red X is the gene that conveys our transgenic phenotype. And it's diploid, but they have a wild type version of X, so it's little x, we'll say. So transgenic, meaning it's got some modified X, and it's also got one copy of a wild type X because it's diploid, okay? Now, let's imagine a scenario where this transgenic X fly, it's heterozygous. Okay, if it was homozygous, it would have a double copy, but let's imagine a scenario where it's heterozygous. It has a wild type copy of the allele, and it's got a transgenic copy of some modified allele that has some kind of an enhanced functionality. And let's say this Joe mates with this Jessie, a female, and she's wild type, completely wild type. Okay, what's going to be the result of their offspring? We can just map out this Punnett square. This is due to meiosis. The offspring will look like this. And let's now envision the outcome of this mating. So in this next generation, from this mating, 50% will still be transgenic, but 50% will have reverted to wild type. Okay. And so this is already a problem in a sense that we're every generation, we might be losing an amount of our population that's transgenic if we're trying to get this thing to establish itself in a population. In each subsequent generation, 50% might revert to the wild type. Okay. Not only that, but let's emphasize now what we discussed prior, where if you have the transgenic X, you have a fitness disadvantage. That means these individuals are more likely, in theory, either to die or they're going to produce less offspring than these ones. So there's going to be an amplifying effect, and eventually you might see the complete disappearance of this transgene X, okay, if you're not selecting for it. So you might ask the question, well, how do we even make transgenic X then? How do we even keep transgenic flies if it's just destined to sort of just disappear over time? How do we even make these and keep these in the lab? Well, we implement artificial selection. And we've talked about this a lot in terms of like eye collar. So what we do with flies is we mate them in a tube, and we induce artificial selection on this, what would be this Punnett square. And we throw these ones away. We kill the wild type ones away because these ones will have red eyes. And we only let the red eyes mate in the next generation. So let's draw the result of that next generation cross then, what we do in the lab. The result is that now if we are selecting, 3/4 are transgenic, and one of those 3 is homozygous, 2 are heterozygous, and 1/4 is wild type. Now imagine we do this again and we kill these ones. We kill these ones, okay? Now in the next generation, more and more and more are going to be becoming homozygous, and eventually we're going to get a population that is just looks like this. And all the offspring are homozygous. And by this point, we can relax the selection in the test tube, and we don't have to pick anymore because they're all that type. So what I've drawn out for you here is sort of like the artificial selection means of sort of generating a kind of like gene drive. Like I wanted to build a population that was 100% this transgene X homozygous, and so I just used selection to kill off ones that were wild type, and eventually I got a population that was 100% homozygous for my transgene. Now that's not really gene drive, but it's sort of giving you like the effect. The effect is that I've converted a population into my desired population. Except in this case, I was using artificial selection to get what I wanted. Now, in the wild, it's very difficult for us to select out the things that we want to select out or to select for. It's very difficult for us to do that in a wild population. So we have to develop genetic structures which are able to sort of fix or hack into this sort of Mendelian ratios. We need to break the Mendelian ratios such that we can have an organism that sort of always passes on the gene that we want. So let's now kind of give like a formal definition of gene drive. And I'm going to say that this is a genetic mechanism that I'll say breaks, because I think that's a fun term, breaks Mendelian genetics in, this is a fun definition, in your favor. And I'm saying like your favor as you are the scientist. So let's draw what this looks like, okay? Let's imagine a scenario where we now have a gene drive. We got a gene drive fly. It's green. Okay. And we have a population of wild flies. I can just each of these flies now that I'm expanding on is a dot. Okay. Now we want to replace this black population of flies with the green population of flies. So in a gene drive, you release this one into the population, and the next generation looks like this. Let's say this is the F1, this is the F0, and the next generation looks like this. F2. And the next generation looks like this. They're all transgenic green for our modified insect. That would be gene drive. How can we do that? How can we create genetic systems that do this? So let's now attempt to draw a gene drive genetic circuit. And there was a paper on this, and they called it the CRISPR chain reaction. Chain reactions are holy in science. The nuclear bomb is a chain reaction. Chain reactions are special. Chain reactions are things that amplify themselves and don't stop. They can't be controlled. Those are chain reactions. So chain reactions are special in science. They're fun to think about. So let's draw a CRISPR chain reactor. Okay, so imagine now we're in a fly. And the fly is just a model for any insect. You can imagine that we're in any insect, but let's say we're in a fly. And let's say we are targeting gene X, and we want to drive a very specific modified gene X, where we have some kind of modification, which changes gene X to do something that we want, okay? So I'm going to draw the transgenic gene X like this. And wild type gene X looks like this, okay? So now imagine a scenario where we want to create a gene driving transgenic gene X. How do we do that? Well, let's start with CRISPR, okay? What do we need for CRISPR to work? We need a Cas9, right? That's the nuclease. And we need guide RNA. And we are modifying gene X. So we're going to follow the pathway of homology-directed repair. And so we're going to need a template of gene X. Actually, I should redraw this in the transgenic gene X. We need a template for that. These are the three things we're going to need to build a special circuit that is going to drive a chain reaction, converting all gene X wild type in a population to transgenic gene X. And we have to organize them in a special way for this to work. Our guide RNA is going to have to target gene X to make that modification. So the guide RNA is going to be conveying the mod. It's going to be attacking the site where we want to insert the modification. So that homology would be built into that guide RNA targeting sequence, and it would match the site where we want to make the modification. Okay. So that's the first thing we need in our genetic circuit is we need to put in an ORF, that is our guide RNA, with a promoter that targets this little region right here. Let's say that's the targeting region. This region targets gene X, where we want to modify, right there. Now, the second thing we need, or the first thing we need, so we got this. The first thing now we need is we need an enzyme, an ORF, that codes for the Cas9 with a promoter. Okay, so let's build that into our genetic circuit. Got it. Number one, checked. We got it. Now, the third thing we're going to need is to provide the template for homology-directed repair. But we're going to do this in a special way. We're going to have two homology arms. Let's say homology arm 1 and homology arm 2 over here. So over here, on the 2 side, let's put a homology arm that is our modified gene X exactly. Okay? So this is what we call homology arm 2. And let's put a homology arm over here, homology arm. That means it's matching sequence. And let's say the homology arm 1 is somewhere 5' of gene X. It's somewhere to the left of gene X. Let's say maybe it's the promoter region. Or no, we wouldn't want to disrupt the promoter. Let's just say it's far 5'. 5'. That means in the actual fly, what this means is that in the actual fly, it's got a wild type gene X with a promoter. Here's the 3' side, and the 5' side of that gene X, is over here. So what I'm saying is let's take this region and put this region, matching sequence, right there in our CRISPR construct. And let's take this gene, except let's do our modifications in a plasmid beforehand. We'll modify it to how we want it, and let's put that modified region right here. Okay? So this is what our special construct looks like. I'll draw that below and I'll reiterate what it is. Okay, so we're targeting this gene X, and we put a 5' homology arm here. We had a gene sequence, which was the Cas9 ORF. We had a guide RNA ORF with a promoter, and that targeted gene X. The guide RNA targeted that. And then we actually took this, a wild type gene X. We did this prior. We cloned it into a plasmid. We modified it by either site-directed mutagenesis, or we did some kind of nucleic acid manipulation, and we modified it such that the insert was no longer wild type. It was a special gene X, which is what we were trying to drive. And then we took this and we popped that right here. And it has a promoter. Now, let's think about what's going to happen when we insert this entire big construct. Insert this into Drosophila, a transgenic insect. Let's insert this whole thing into a transgenic insect. Okay, so we pop this thing in. Now what's going to happen? Okay, let's just think it through with the central dogma. Cas9 is going to get transcribed into messenger RNA and then translated into Cas9 protein. Remember, in order for CRISPR to work, it's got to complex and form that RNP. So then what happens is the guide RNA is going to be transcribed, and that guide RNA will complex with the Cas9. This is going to form the RNP, and it's going to have a little targeting region. And remember how I said that the targeting region is going to target gene X. Okay? Now what's going to happen? We're in this fly. This is all inside of this fly here. Okay? And this fly has 2 wild-type copies of gene X in its chromosomes. Okay, it's diploid, so it's got 2 chromosomes. And on those chromosomes somewhere is a sequence of the gene X, which we're targeting. And it's also got this insert, so it's also got this transgenic copy. It's got this whole thing inserted. So somewhere on its chromosome, it has this giant whole thing inserted. So it's also got 1 extra copy of gene X, which is the modified version, which is that version right here. Okay, because we inserted that, remember? So now let's think through what's going to happen. This Cas9 complexed with the guide RNA, which is targeting gene X, is going to come find the wild type gene X's on the chromosomes. Okay, so let's expand this region on the chromosomes. Let's say the gene X, the wild type gene X, is right here. This RNP now is going to find this sequence and it's going to cut it. It's going to induce the double-stranded break, which CRISPR is famous for. It's going to cut the DNA. And this chromosome is going to look for a template to repair itself with, right? Okay. And what is it going to find? It's going to find this construct, which we inserted as a template for homology. So what's going to happen then is this chromosome is going to recombine through homology-directed repair, and the resulting product is going to be a chromosome, and on that arm where gene X once stood is going to be our entire sequence. Okay, it's going to be the homology arm 1, which we chose 5'. It's going to be the Cas9. It's going to be the guide RNA. And it's going to be our modified gene X. Okay. Now the process is going to repeat because these genes are on the chromosome. Okay? They're on the chromosome. So what's going to happen now is, now what we have is we have one chromosome converted. But there's a second chromosome because it's diploid, okay? So there's a second chromosome that has still a wild-type gene X left. Well, what's going to happen? The same thing's going to repeat. The Cas9 gene is going to get transcribed and made into Cas9 protein. The guide RNA is going to get made, and that guide RNA still targets the same thing. It's targeting gene X. Okay? So this RNP is going to now do the same thing. It's going to seek out the gene X. It's going to cut it, induce a double-stranded break, and the same thing is going to happen again. And it's going to find a template for homology right here. And now what's going to happen is you're going to have 2 converted chromosomes to gene X. Okay, so we've essentially taken an organism that was originally heterozygous for a transgene and converted it to homozygous. Now all of its children are going to inherit one of these transgenic chromosomes, and the same process is going to repeat such that all offspring of this chromosome and this chromosome are going to produce, with 100% efficiency, chromosomes that look exactly like it, because they're just going to, if they find a wild-type chromosome, they're just going to convert it instantly. Okay, so this is a gene drive because all of its offspring are going to inherit this genetic construct because of that conversion. So this is what's called a CRISPR chain reaction. Now you might have a question, which is a question that I also had, which is, well, how does this RNP know to target the wild-type gene X and not the transgenic gene X? And the answer is that it doesn't necessarily know. But if it hits itself, if it hits its own ORF, again, if it cuts its own ORF, it's just going to repair with itself. So there isn't going to be, it's just going to sort of maintain itself. It's going to maintain itself if it attacks its own ORF. But if it attacks the wild-type gene X, it's going to go through this conversion process. And it's eventually going to convert because the transgenic one is going to maintain itself and the other one is going to be converted. So it doesn't matter, in the long run, it's going to target both. And it's all going to be converted in the end. Okay, so it's been demonstrated that in the lab, this type of converting CRISPR chain reaction works. It works in the lab. But the lab is different from the wild. So there's a question of, will this work in the wild? And they have been trying to implement in field studies, in I guess small-scale field studies, some of these CRISPR-Cas9 chain reaction gene drives. And there's a question of, is it going to work? And spoiler alert, the answer is that as of right now, these systems don't seem to work in the wild. They work in the lab, but they don't necessarily work in the wild. And let's talk about some of the reasons why they don't work in the wild. One of the reasons is that expressing this guide RNA, this Cas9, and all these having these transgenic homology arms and converting things, that is, again, goes back to fitness. A lot of times it's going to reduce the fitness of the organism. And the organism actually does have an alternative. So let's describe the alternative scenario, which I'm going to be calling the resistance to the CRISPR gene drive. Let's talk about how resistance evolves. So let's just draw it out again. Our construct is a homology arm 5' 1, our Cas9, our guide RNA, which targets gene X, and our homology arm 2, which is in fact a transgenic gene X. Let's talk about how resistance evolves to the spreading of this construct. And it's all based on whether or not the cell decides to do homology-directed repair or blunt end repair. Okay? So now let's draw out again what's happening, but under a different scenario that can happen. Here's gene X on the chromosome. Okay? Cas9 gets made into protein. Cas9. Guide RNA complexes with the Cas9. It's targeting gene X. And it cuts it at gene X. Now the cell has a choice, like I described in the last lecture. Does it do homology-directed repair, or does it do blunt end repair? It has to ask itself that question, and there's a decision tree. And if it goes the route of homology-directed repair, it will convert this ORF into an ORF that looks like this construct. But if it chooses blunt end repair, remember what happens in blunt end repair. So this is gene X, which was cut. If it does blunt end repair, it's going to take this and smash it together. And sometimes when blunt repair occurs, some things get left out. Maybe an A or a T or a G falls out. And there's a disrupted, let's say it's a disrupted gene X as a result. Okay. A disrupted gene X. Now let me ask a key crucial question. Can the guide RNA, which cuts at this site specifically, and the guide RNA is only targeted to that region if the base pairs match precisely. Now, what I just told you is if the cell chooses blunt end repair, there's a statistical chance that gene X comes out mutated, comes out broken in such a way that the guide RNA might not no longer recognize gene X. Because if the site that it's targeted has now been mutated, the guide RNA can no longer find gene X. So when I draw these dots and it's targeting, that can no longer happen if the site is mutated. So essentially what I'm saying is, in a nutshell, the organism is sort of conveyed with a biological choice. One choice is convert gene X to that big long transgenic insert. Convert the gene X to the insert. Or a second choice is mutate gene X such that it's no longer targeted by the guide RNA. The guide RNA no longer matches the sequence. And when I say choice, I'm saying this very liberally. I mean, this is all this, the organism and the cells are not making a conscious choice. They're doing one or the other, and then selection works on these choices and kills off the ones that aren't the most optimal ones. And what happens is this second choice seems to usually be the most optimal. It seems to usually mutate gene X in a way such that it can no longer be targeted by the guide RNA. And then it sort of just rids itself from this entire system by evolving resistance. So one of the problems to the CRISPR-Cas9 and implementation of the CRISPR-Cas9 gene drives is that by their very nature they are nucleases. And they're homing nucleases. And the things that they target can mutate such that they can no longer be targeted. Okay? So it's like they induce their own resistance because the thing they target, they cut up, and that process of cutting it up and repairing it is instigating mutation in that site. So CRISPR-Cas9 gene drives catalyze resistance to themselves. So this is one downside of this system, and a lot of the research now is based on understanding how this resistance evolves and sort of understanding it as a system of almost like car brakes, like brakes. Like people are researching this to see how these brakes happen, if they can sort of disrupt the brakes in a way that then they can create a gene drive that continuously spreads. And then there's also people on the other side who are like, that's a terrible idea. We do not want to invent a car without any brakes, because imagine what that's going to do to the environment if something goes wrong. So there's a lot of people now researching the evolution of resistance to these CRISPR-Cas9 nuclease chain reactions. Now let me briefly just discuss some other sort of variants of gene drive. Okay, there could be behavioral gene drive. And let me give you a scenario for this. So imagine there's a mosquito, and this is I guess what you could say a proven scenario of behavioral gene drive. Imagine there's this mosquito, and in that mosquito, in its gut, there is a bacteria called Serratia. And we'll call this Serratia strain AS1. Okay. And every time that insect lays eggs, it tends to like crap out some Serratia on its eggs. Okay. And mosquitoes lay eggs in the water. So imagine these are in the water of a pond or a tire. Those bacteria are now going to spread out into that pond. Okay. Now imagine the eggs hatch, and what comes out are little larvae that look kind of like this. Okay. And these larvae swim around in the water and they eat some of the bacteria. The bacteria now get into the gut of that larvae. And this larvae now progresses through normal development. It becomes a pupae, which kind of looks like this. And the pupae now becomes an adult mosquito. And the bacteria are still inside of its gut. And now when this mosquito lays eggs, the same process happens. And it craps out some of the bacteria onto the eggs in the water. Now imagine you are an uninfected larvae swimming around in this water, which is invaded by this bacteria called Serratia. You're eventually going to eat this larvae, and it's going to infect you. So this process of kind of like behavioral infection is going to perpetuate itself until the population is 100% infected with this Serratia. So it's almost like a disease epidemiology. It's almost like the spreading of a disease. But when it becomes gene drive, is if, imagine the Serratia was a modified Serratia. Imagine we took the Serratia and we brought it into the lab and we cultured it on petri dishes and we transformed it with a special plasmid or a special insert of a gene X. And let's imagine that that gene X killed malaria, which spreads inside of the gut of mosquitoes. Now all of a sudden what we're going to have is we're going to have an organism which spreads itself through the behavioral biology of the mosquito, and it gets into all the guts of all the mosquitoes. And you can imagine now, if that mosquito picks up malaria, which will be green, if it takes a blood meal that's infected with malaria, it's also got this Serratia in the gut, which has the plasmid or the insert of gene X, which is now its function we programmed to kill malaria. Thus, in such a system as this, and this system was built and described by Marcelo Jacobs-Lorena's lab at Johns Hopkins, this system is going to be a system of behavioral gene drive, which is driving a favorable phenotype in a symbiont. It's a paratransgenesis strategy compiled with the gene drive strategy. Okay? This was in a science paper. So this is an example of a behavioral gene drive. Okay, so I hope this was a sufficient discussion of gene drive systems. The CRISPR-Cas9 gene drive chain reaction is a complicated genetic circuit. If you don't understand it or if you want more clarity, this is the paper, The Mutagenic Chain Reaction, a method for converting heterozygous to homozygous mutations. And it's by Valentino Gantz and Ethan Beer. Okay, so you can go to this paper and you can read all about it right here, and it's free on PubMed. But I think the general concept is that you build homology arms flanking a Cas9 and a guide RNA. And if the guide RNA is targeting what one of your homology arms matches, you can get a chain reaction that converts and converts and converts and converts until you convert everything in a population to homozygous, assuming that the resistance doesn't happen. Okay? I hope you guys have a great day.